Okay, so we've been working with this Clec9A targeting platform, which actually has been developed by my longtime collaborator called Mireille Lahoud from Monash University. And what Mireille did was to, okay, is that one? Yeah, to isolate and engineer a monoclonal antibody that recognizes the C-type lectin receptor Clec9A that is almost exclusively expressed by the CDC1 subset in mice, which has its equivalent in human as well. So what Mireille does is to fuse genetically the vaccine antigen candidate at the heavy chain of this monoclonal antibody. And this antibody construct will directly bind to the Clec9A receptor and will be internalized. And once internalized, this construct is going to undergo 3 different fates. The first one is cross-presentation, because the CDC1 are excellent cross-presenter, and this will give you an MHC class 1 antigen presentation and a strong CD8 response. The second fate of this construct is to undergo the lysosomal pathway and being degraded into the lysosome, leading to MHC class 2 antigen presentation and therefore a CD4 response. And finally, this construct can also go through the recycling endosomal pathway and be re-expressed intact at the surface of the cell, and now allowing the B cell to interact with their B cell epitope. So basically, when you use this strategy, you really plug your vaccine antigen candidate into an optimal immunological environment for inducing a very strong humoral and cellular immune response. So we have been leveraging on this platform and using vaccine antigen candidate against infectious disease. And before the pandemic, we have published this first paper showing that when we fuse 3 copies of this 23 amino acid peptide called M2E, which is known to be a universal influenza A vaccine candidate, we were able to show that a single systemic administration was enough to induce a very sustained and high titer of antibody against M2E that were protective. So of course, when in 2020, when the COVID-19 hit our shores, what Mireille did very rapidly was to fuse the receptor binding domain from the SARS-CoV-2. At that time, we had only the ancestral sequence. And she made this Clec9A RBD construct. And once again, we showed that a single systemic administration of this Clec9A RBD construct was sufficient to induce a very sustained antibody response that was neutralizing, able to protect against a challenge. And importantly, we could see a prolonged affinity maturation upon a single administration and cross-reactive T-cell response. So all well and good, but 4 years, fast forward 4 years, majority of the population is already now has some form of immunity against SARS-CoV-2, whether it's through vaccination or through infection. So we were thinking of repositioning this Clec9A RBD construct as a booster vaccine in a population which has some sort of SARS-CoV-2 pre-existing immunity. And what we did was to benchmark our Clec9A RBD construct to what was available at that time as a booster vaccine, which was the bivalent mRNA Pfizer vaccine, which expressed the spike from ancestral and Omicron BA.5.4.5. So why do we want to do this? Because we know that the mRNA vaccine have a number of shortcomings, and we wanted to see whether our Clec9A RBD could actually address at least some of those shortcomings, which are antibody titer waning. It doesn't protect against the other clades. It's mainly limited to the clade 1b and mostly the SARS-CoV-2 family. And also because it's given intramuscularly, we know that mRNA vaccine do not really confer a strong mucosal immunity and therefore does not stop transmission. So that's what we are trying to achieve. These are the desirable attributes of this vaccine booster, which will be able to protect against clade 1b, but also clade 1a, which is, clade 1a, which is where SARS-CoV-1 belongs to. And we would like to induce respiratory immunity and also a durable response. So this is the experimental design that we have used throughout. And basically, the mice received intramuscularly at 3 weeks interval the original Comirnaty Pfizer vaccine, which expressed the ancestral spike. And then we wait for 3 months. So we wait that the antibody titer waned a bit. And 3 months after the second dose, we now come with our booster. And the booster will be either another intramuscular injection of this bivalent mRNA vaccine, or we boost subcutaneously with one of our Clec9A RBD construct. And this time we use an RBD that comes from Omicron XBB. And we also use a SARS-CoV-1 RBD as a booster as well. And what we did was to measure, to bleed these mice at 1 month and 6 months post-boost, and using a surrogate neutralizing assay developed by Dr. Tan-Chi-Wa, who was at that time under Prof. Lin-Fa Wang lab, but now he's an independent PI at NUS. So he helped us to look at the neutralizing activity of the immune serum samples. So these are the results. In red are the control animals which were boosted with the bivalent mRNA vaccine, and in green are the animals that are boosted with the Clec9A XBB construct. So you can see that the neutralizing activity, whether it's pre-Omicron, Omicron, Clade1B, and Clade1A, what we see is clearly the decrease in the antibody titers in your mice boosted with the mRNA bivalent. In contrast, when you boost with the Clec9A XBB, you don't see any waning effect. So at 1 month and 6 months, the antibody titers are sustained. And what we see is that we have neutralizing activity against the whole Clade1B, but unfortunately, we don't see much of a neutralizing activity against Clade1A. So when we now boosted the mice, instead of the Clec9A XBB, we had the Clec9A CoV1. What we saw was that now we had a cross-clade neutralizing activity. So now we were able to have also a neutralizing activity against the Clade1A. However, very surprisingly, we saw that when we boosted with Clec9A CoV1, we had a decrease in the antibody titer over time. So that was quite unexpected. So that means that we are dealing with something that is quite suboptimal, because if we boost with the XBB construct, we have a nicely sustained antibody response, but it's not cross-clade. And if we boost with the CoV1 RBD construct, we have a nice cross-clade, but it's not sustained. So what did we do? We just decided to combine both constructs together and came up with this formulation that we called the Clec9A RBD Omni, hoping that it will do everything. So what we did, again, we repeated the same experimental approach, and this time we have 2 groups. One, again, our control group, which is boosted with our bivalent mRNA vaccine, and the other one that is boosted with the Clec9A Omni formulation. In addition to look at the neutralizing activity in the immune serum sample, we also challenged those mice with Omicron BA1. So looking at the neutralizing antibody titers, here is the titers against the pre-Omicron and Omicron series. You see again that the neutralizing antibody titers in the mice that were boosted with the Clec9A Omni construct are not only significantly higher than the titer in the control group, but again, they are sustained over time. And we see the same picture against the other Clade1B series, and again, the Clade1A now, which is not seen with the control, the bivalent mRNA vaccine. So we concluded that now we were able that when boosting with the Clec9A RBD Omni construct, we could now achieve a sustained and a cross-clade neutralizing antibody response. So when it comes to the challenge, we did the challenge at 1 month post-boost, and we could see that there was a reduction compared to the non-immunized mice, which was equally good for both groups. However, when we did the challenge at 6 months post-boost, we could see that now the mice that were boosted with the Clec9A Omni construct had a lower viral titer compared to the mRNA boosted group, and that is due to probably the waning antibody titers in these mice. So we wanted to also explore the nasal route, because one of the shortcomings for the mRNA vaccine, remember, is that they don't really induce a strong mucosal immunity because they are given systemically. So if we give this, now this Clec9A Omni formulation via the nasal route, are we going to be able to trigger a nice mucosal immunity? So we delivered this two construct via nasal drop. And again, we looked at the neutralizing activity, not only in the serum, but also in the bronchoalveolar lavage and the nasal laval fluid. And again, we challenged those mice. So what did we see? Looking at the neutralizing antibody titers inside the serum, and we have a representative here of pre-Omicron, Omicron, and Clade 1A. So we saw in the serum that, again, just like the systemic booster, very sustained and strong neutralizing antibody titers in the serum of those mice, which was greater than the mRNA bivalent boosted mice. And very nicely, we saw that the neutralizing activity in the bronchoalveolar lavage and nasal laval fluid was very significant, whereas they were not detected or barely detected for the control group. So we were also able to detect IgA, RBD-specific IgA, in those bronchoalveolar lavage and nasal lavage fluid. And because I don't have a lot of time, but I just want to highlight a few of the in-depth characterization of the immune response that Nicolas did in this work. And one of the very exciting findings was the ability to detect what we call a triple cross-reactive B-cell subset, which is in blue here. Basically, these are B-cells that are able to bind to the three types of RBDs, from ancestral, Omicron, and SARS-CoV-1. And we were able to detect those subsets uniquely in the mice boosted with the Clec9A Omni formulation, not in the control group, and in the spleen, the lung, and the nasal tissue. And at 6 months post-boost, meaning that it's really a very sustained response. So concomitantly, we were able to detect also RBD-specific follicular helper T cells in the spleen and in the lungs of those mice at a much greater frequency compared to the control group. And this was correlated with the detection of higher proportion of RBD-specific germinocenter B cells in those spleen and lungs, and all of these were detected at 6 months post-boost. And we were also able to see in the bone marrow that there was a greater proportion of long-lived plasma cells versus non-long-lived plasma cells in those mice boosted with the Omni construct. So all of this points to the fact that when you boost with this Omni construct, the immune cells undergo an extensive affinity maturation, and we believe that these triple cross-reactive B cells are likely to produce cross-clade neutralizing antibodies. So we look also at the T cell response, and again, to cut a long story short, basically the T cell response is highly cross-reactive in these mice boosted with the Omni construct, and they are also polyfunctional. So the magnitude of the response when we restimulate with the different overlapping peptide is significantly greater than what we see in the control mice, and the T cell response is even more prominent in the lung and the nasal tissue, which is not unexpected, given that the Omni construct was given nasally. So for the challenge, similarly, we see that we have an equally good, similar, comparable protection in both groups when the challenge is done at 1 month post-boost, but we see a greater, a lower viral titer in the mice that are boosted with the Omni construct when the challenge is done at 6 months post-boost. So clearly, when we boost nasally with this Clec9A RBD Omni, we see a superior protection when the challenge is done at 6 months. So to conclude, I hope that this data convince you that this Clec9A RBD Omni potentially can really achieve and fulfill those desirable attributes to compensate, to address the limitation of mRNA vaccine, which is when it comes to sustainability of the antibody titers. Now we are able to induce a cross-clade response against Clade-1A and 1B, and we have shown that it leads to significant protection. Very excitingly, we are able to see that for the first time this Clec9A delivery system is amenable for the nasal route of administration, and it doesn't compromise the systemic immune response. The detection of those triple cross-reactive RBD-specific B cells potentially means that those B cells are now producing cross-clade neutralizing antibodies. So this platform is really a plug-and-go platform that is highly suitable for pandemic response. We don't believe that GMP manufacturing will be a bottleneck because it's just an antibody, and the manufacturing process for antibodies is very mature and available everywhere in the world. So now the $1 million question is whether it's going to translate to human, and although we don't have the answer now, we are very hopeful that it will, for several reasons. The first one is that, as I told you at the beginning of my presentation, the CDC1 has an equivalent in human. It's this disease, CD141 positive disease. A prior study showed that this disease subset is actually can be detected in majority of our tissues at a constant level throughout our lifespan. Now Mireille has engineered fully humanized Clec9A monoclonal antibodies that recognize human and also non-human primate Clec9A receptor, and she has generated this Clec9A RBD construct, and in a couple of days we are going to start our immunogenicity in non-human primates. So we are very excited, and hopefully I will be able to report very good data maybe in half a year time. So I'm going to stop here. I would like to thank the people who have worked on this project, in particular my very talented PhD student, Nicolas, who generated all the data that I showed today, our collaborator, Paul McCary, Chiwa, and of course, Mireille and her team, who has been really the mastermind of this platform. This work is supported by the Prepare funding agency. Thank you for your attention.
Hi, Sylvie. Thanks so much for your excellent talk. It's such a clever way to bring the antigen to the correct cells. So I'm very excited about this platform. And I was wondering, in terms of the limitations surrounding the kind of molecule that you can attach to the Clec9A antibody, because here I see you've used RBD and not the host spike, so and then had to combine the variants. So is there a limitation?
Yeah, I mean, obviously you can imagine that for your antibody to remain stable, you're going to have some sort of a size limit or conformation limit. So indeed, Mireille tried to fuse the whole spike initially. It just couldn't work. So she couldn't produce the antibody. The antibody was not stable. So I would say that yes, there is a size limit. But I think the power of this platform is really for small, weakly immunogenic antigen. And our very first story was with this M2E vaccine candidate, which is a 23 amino acid peptide. The problem is that it's weakly immunogenic. So you need to find a way to deliver this little peptide and make it now seen by the immune system. And I think that this platform does just that. So I think the potency of this platform is to make a weakly immunogenic antigen, and we are talking also about cancer antigen, for example, which are usually small peptide. So you can give them a much stronger immunogenicity using this delivery system.
Okay. Another maybe naive question, right? So you're giving it to the DC, but where does the adjuvant signal 2 come from?
So there is an adjuvant. I didn't mention this.
I see.
It's
Okay.
poly-IC. Okay. And poly-IC is, we tried a few, and we found that poly-IC was the best by far. And the reason is because the CDC subset produce a lot of TLR3.
TLR3, okay.
So it's not surprising that poly-IC is one of the best adjuvant.
Okay, thanks.
Thank you.